So as many of us know, being the last speaker at a big symposium is challenging. And so I was trying to think of some way of jazzing up my introduction to get people alive. And I sort of realized that our first speaker's name started with R, and the last speaker's name started with R, and I started with R. Could I take that and make it into something that was interesting? And I couldn't think of anything. So therefore, I have to go back to sort of a standard introduction, okay? And so that I'm not going to introduce Dr. Richard O'Reilly, whose picture should hopefully appear someplace up there. So and to be politically correct, I view that Dr. O'Reilly and Dr. Reiser are the godparents, not the godfather anymore, but the godparents of haploidentical transplantation. And as Rupert mentioned, that Yair developed the technology for T-cell depletion. Dr. O'Reilly was responsible for clinical transplantation. And in 1981, they announced that they had done a successful T-cell depleted haploidentical transplant. And that was the beginning of haploidentical transplantation, which is this meeting is was the precursor of this meeting. But one of the things that's important to realize at that time, Dr. O'Reilly had been involved in stem cell transplantation for more than a decade. So in 1968, when Dr. O'Reilly was a pediatric resident at the University of Minnesota, David Kemp was admitted. And for those you know, David Kemp was the person that Robert Good successfully transplanted. So the first successful allogeneic transplant occurred at Minnesota, and Dr. O'Reilly was part of the team there responsible for the care of him. So in a sense, Dr. O'Reilly was there really at the beginning of allogeneic transplantation. Once Dr. O'Reilly finished his pediatric residency at Minnesota, he went to the CDC, and then he came to the Children's Hospital in Boston as an infectious disease fellow, and that's where he and I first met. And there he worked on the development of the vaccine for Haemophilus influenza that ended up being the Hib vaccine. Well, during that period of time, Dr. Good moved from Minnesota to become the head of Memorial Sloan Kettering and called Richard and said, since he was at Memorial Sloan Kettering, he wanted to start a stem cell transplant program, and would Richard come down to run that program? And as they say, the rest is history. And so Richard had run the program at Memorial for probably more than 50 years. And I'm not going to, quote, bore you all by giving you a litany of all the achievements Richard did in those 50 years. But what I thought was important to say something about not what Richard had done in terms of the science and clinical, there he is, but what his contribution has been really to the field of at least stem cell transplantation in North America. So in the early 1990s, a lot of us felt that the stem cell transplantation was really not being supported well by the American Society of Hematology, and that what we needed was a society that would be focused on the needs of stem cell transplantation. And the American Society for Blood and Marrow Transplantation, or ASBMT, was established. And at that time, we realized for that society to have both scientific and academic credibility, we needed to have a journal. And we started, BBMT, or the Biology of Blood and Marrow Transplantation. And for the first 10 years, Dr. O'Reilly and Dr. Bloomer was here, were the editors of that journal. And without their commitment for that first 10 years, the journal and the society really would have never gotten off the ground. And so I think that contribution to the field of transplantation is one of Dr. O'Reilly's unique contributions. I do want to go back now to at least one of his scientific observations, which was the observation that if you gave mature donor T cells to a patient who'd had a transplant and had EBV-induced lymphomas, that the introduction of those mature T cells could result in the cure of the EBV-associated lymphomas, demonstrating that mature donor T cells had both anti-lymphoma and anti-EBV activity. And that proof of principle, in my mind, is the basis for the present work on CAR T cells for treatment of malignancy diseases, and for the interest in the issue of virus-specific T cells, which is the subject of his talk today. Dr. O'Reilly.
Thank you a lot, Robbie. You're spectacular in terms of making me look good, particularly at the end of the day. I apologize for not being there myself because you're at a wonderful place that I absolutely love. I want to, however, go quickly through some of these studies. And I think one of the thoughts I had when I talked about this early on with Alice and also with Robbie, I mean, it has been my dream with the T-depleted transplants to be in a circumstance where I could, in fact, give T-depleted grafts and then give T cells immediately. The commercial aspects of that are really kind of difficult, but what I am going to talk about are the various methods that have now been developed. But most importantly, I'm going to talk about our own experience in terms of what we can do, and in particular, talk about some of the areas in which we've had failures. And the reason why I say that is because these viruses are very smart. And it's important for everybody to understand that they come up and they can come up really fast. So I have to tell you that Atara Biotherapeutics licensed the EBV specific T cells. For those of you from Europe, you know that EMA has approved these cells. They're in the final stages of being looked at also by the FDA. But we have a bank of around 330 of these lines that were transferred. Now, what I'm going to do briefly is note that early in the transplant realm, it was pretty clear that if you went outside the context of an HLA match related or unrelated individual, the concern for acute graft versus host disease of a lethal or severe variety was very high. And it was clearly Rainer Storb in particular, who was so key in the development of a series of drug prophylaxis methods to deal with minor alloantigen specific T cells in the context of a matched marrow transplant. But even there, as you can see, the incidence of grade 2 to 4 graft versus host disease was quite significant. And most importantly, the incidence of chronic graft versus host disease ranged from about 30%, usually up to 50%. The obvious other concern was that one could not go beyond the matching. There were some isolated instances of young kids who received HLA disparate transplants for A or for B or for A plus B, as long as they were MLC compatible, but that was basically it. So we set about developing an approach. Yair Reisner and I developed this SPA negative, E negative marrow graft. On this slide, what you're seeing is what are the actual doses of T cells that are administered. And as you can see with those, normally when you're talking about a transplant of unmodified marrow transplants, you're talking about doses of 10 to the 7th to 5 times 10 to the 7th at least of T cells per kilogram. With these type of approaches, you could reduce that down to 4.5 times 10 to the 4th per kilo with the lectin, or even lower with the Miltenyi or the Isolex down to 1 to 6 times 10 to the 3rd cells per kilogram. And yet you also saw a very marked saving of the early CD34 positive cells, such that you could give from peripheral blood stem cells doses of anywhere from 5 to 9 or 10, and particularly for a young child. We went through quite a while defining residual host T cells as the effectors of graft rejections, but we also developed approaches which included ATG, specifically before transplant, and that along with myeloablative regimens such as TBI Thio-Cy or TBI-Cy, or the Bumel flu regimen that we developed, have achieved consistent engraftment. The incidence of immune rejection is down 0 to 2%, and the incidence of late graft failures is about 2% at max. I'm going to show you a little bit about some of those studies here. Early on, we did T-depleted transplants at our shop using these types of approaches. And here is a large series of 266 individuals who received transplants for AML in first or second remission. these are all individuals who are older, and you note that the median age is 54. It's important also to note that in Memorial Sloan Kettering Cancer Center, the current median age for our transplants of allogeneic marrow are about 59. It's about 59, irrespective of the type of the transplant that you give. What can also be stated, of course, is that the leukemias that we face on a daily basis are more in the individuals over 65 than less. So this is really a critical aspect of the work that we're trying to do. You can also see that a very significant number of these individuals are matched, unrelated, or mismatched donor-recipient pairings. This is in the context of the percentage and the TBI-based or chemotherapy-based regimen. As you can see, for the AML, it's mostly the chemotherapy-based, particularly in the first remission. Yet you can see for the whole cohort that the likelihood of graft versus host disease, grade 2 to 4, was only 17%. And most importantly, the incidence of chronic GVH was 3%. In this cadre of individuals who were transplanted over 10 years ago now, what we're looking at, nevertheless, is that the disease-free survival for the whole cadre is about 53% for patients transplanted in AML in first or second remission. We now come to clearly the randomized trial that was done by BMT-CTN. And in that trial, as you can see, there were 346 individuals who were randomly assigned either to receive the CD34 selected T-depleted transplant or a graft where you gave post-transplant cyclophosphamide, or you received a conventional transplant. And in this case, it was marrow consistently with TAC and MAC and methotrexate as your post-transplant prophylaxis. The results of those, as you know, were published in the JCO, and they caused quite a stir, particularly caused stirs in my stomach, because what you see is that in real terms, the probability of a chronic graft versus host disease and refractory disease free survival is in blue at the top, and it edges out the Cytoxan post-transplant, but in real terms, as you can see also in the green, I'm sorry. Here. In this one, as you can see in A, the lower one there is the conventional transplant. By the same token, if you look at the overall survival, the lower one here is the CD34 population. That is the T-depleted transplant. The overall survival is right on target with the one we just saw for AML in first or second. This is for a wider spectrum, but nevertheless, it was a disappointing feature of the game. What we found here, again, was that the incidence of severe chronic GVH is the lower blue line. It's the lowest of the crew, and that is clearly significantly better. You also see that the transplant-related mortality, however, is highest in the CD34 population of T-depleted grafts. What we also see, however, and this is really important, is the fact that despite a great deal of hoopla about the idea of the graft versus leukemic response, wherein the idea is that the T cells contributing to GVH are the cells that contribute to resistance to relapse. But that hasn't held now in 2 randomized trials and several other comparative trials. And as you can see, the blue is a little bit lower than the green. That is, the CD34 incidence of relapse is a little bit lower than TAC methotrexate protocol. So what you see is that there's no difference in relapse. The central difference that's contributing to problems in terms of relapse-free survival and survival is the issue of non-relapse mortality. So the major question that we've had for quite a while is, can we now be in a circumstance wherein we could give a T-depleted transplant, abrogate graft versus host disease, yet by the same token now contribute to the elimination of the transplant-related mortality. And that mortality is, in fact, very significantly a part of the mortality associated with reactivation of the latent viruses, EBV, CMV, and adenovirus. So I'm going to talk about two of these, namely EBV, because we have large experience there, as well as with the CMV. As you know, for hematopoietic transplants, unmodified grafts usually are about 1 to 3% incidence, but the T-depleted can be as 5 to 7%. It's actually closer to 5% at the present time. The other question to be raised here is the degree to which ATG, which is administered to our patients before transplant to ensure engraftment, to what degree that also affects that incidence. And that may be a feature coming up now that will make our efforts a lot easier. These are, however, very aggressive, usually clonal, diffuse large B-cell lymphomas. Most importantly, also, these are almost exclusively donor, although they're not always donor. There are several high-risk features, particularly older patients who have ongoing GVH treatment, or individuals with extranodal disease or more than 3 sites. In our group, we're looking only at patients who have failed rituximab and rituximab plus chemotherapy, also in the organ transplant grouping. We introduced the idea of giving lymphocytes unselected back early on, and this was the first instance in which cells were used for EBV in this regard. But at that time, there was a letter that came back to the New England Journal of Medicine from Malcolm Brenner, and he was absolutely right. He said, yes, you could give the DLI from a seropositive individual, but the limitation you're going to get into is that you're going to have GVH. If you give low doses of cells, you could avoid most of the graft versus host disease, but it was correct. Nevertheless, you can now take cells, and clearly the group at Baylor has also championed this, to use these leukocytes, separate out the T cells, create transformed B cells that could then be used for antigen presentation, and then generate T cells in response to irradiated and drug-treated EBV BLCLs to create selective T cells that are specific for EBV. The characteristics of these are really important in terms of how you look at these virus-specific cells, because particularly in when you're looking now at the EBV-specific T cell lines in C, as you can see there, they have very striking activity in terms of EBV CTL precursors, but they have extraordinarily low CTL precursors against allogeneic targets in comparison to the responders or non-responders in this group in the conventional marrow transplant setting. What we have done in CTL precursor analyses is to analyze how many you're actually giving. And if you look at the number of EBV reactive cells in unselected donor lymphocytes, as you can see, you're giving about 17 per million, which is about right, because it's about 1 in 10 to the 4th is the actual concentration, similar to what you see in HLA matched donor recipient pairings receiving conventional transplants, because that's the number of major alloantigen specific T cells that you have. It's about 1 in 10 to the 4th as well. EBV specific cells, on the other hand, as you can see, you radically increase that to 156, or about 80,000 now per kilogram. The alloreactive T cells that you can see in the lower panels are reduced radically in the EBV-specific donor T cells to as few as 5 per 10 to the 6th. When we did this using donor-derived EBV-specific T cells, what we were able to show in that kind of a setting was that we had 19 patients initially treated, and 13 of those achieved a CR, and we had no evidence of GVH in comparison to donor lymphocytes that are unselected, where we had 5 patients with grade 2 to 4 GVH out of 30 people treated. The limitations that you have with these is, however, the timing, because these cells take a long time to grow, both the EBV transformed cells as well as the T cells, and the donor availability, because in many instances, the donor is seronegative, or, for example, a cord blood, or in some instances, unwilling or unable to give. Now, as a result of that, there's been a tremendous amount of effort now in the development of approaches that are easier and generate lots of cells. One is still extended in vitro culture because of its advantages in terms of a lack of alloreactive cells. And you can stimulate them with autologous BLCLs, or you can stimulate them with dendritic cells that are loaded with peptides or are transduced to express antigens of EBV. The advantage here is that the yield is quite high. The GVH risk is very low because you deplete the alloreactive cells, and there's HLA restriction, but it varies very significantly with multiple viruses, and I'll show that in a minute. You can also do short-term sensitization with autologous dendritic cells, and then separate these cells using immunomagnetic absorption of either interferon gamma producing cells or cells that express an activating antigen. Again, the limitation here with the short term is its low to moderate yield. The GVH risk is low, however, because it can be up to moderate because you oftentimes will also pull in alloreactive cells in the population that you're getting. And the restriction again varies by virus if it's polyviral in type. The direct isolation from peripheral blood mononuclear cells by binding to multimers, on the other hand, the yield is very low, but the GVH risk is extremely low. And the limitation is you have only one epitope that is targeted in terms of the HLA restriction. There's a new area which is really now coming to the fore, namely using EBV-specific T cell receptors or virus-specific T cell receptors that are inserted into non-specifically activated T cells. Here, the yield is very high, but there is a moderate risk of GVH because of the concern that you're non-specifically activating potentially alloreactive cells as well. The other limitation, of course, is that these cells are restricted to a single epitope and HLA. Now, what do I mean by this variability? If you now take cells from an individual and stimulate that individual, antigen-presenting cells, for example, dendritic cells, with a pool of peptides from a variety of different viruses, what you will have will be T cell responses that are directed against these antigens expressed on the surface of the cell. The limitation, of course, is, for example, if this donor is top A1B8 DR3 and A2B7 DR4, in that setting, B7 is such a hierarchical player in terms of CMV that if you now use the peptides of CMV PP65, the cells will be invariably reactive and restricted by B7 and reactive against 2 epitopes that are presented by B7. And in EBV, you will then find AO201, and there is active competition between these two that really becomes problematic. Adenovirus, less so because more CD4 cell populations are generated in the adenovirus culture. But nevertheless, the one limitation you have with the pooled viral peptides or the pooled viral approach, while it really is something that, as Ann Lean has shown, is extraordinarily promising, it's limiting if you don't know what the HLA restrictions are because they may be different for each virus. Nevertheless, a very large number of cases now have been done using transplant donor-derived EBV BLCLs, and you can see that the responses in these groups range from anywhere from 70 to almost 100%, but the group sizes are small because of the fact getting these cells to be able to do second leukapheresis, or more importantly, generating the cells in the time that is usually allowed you by these diseases, which are oftentimes very rapid, is really quite limiting. The other approach could be then what we developed, which is a bank of EBV-specific T cells and T cell lines. And here we have cells from donors who were marrow donors, but they've consented also for their leukocytes to be used for third-party use. You can generate these cells so that they're defined in terms of HLA at high resolution. They're also defined in terms of virus specificity and HLA restriction, and in most instances now at the epitope level. The other important point is if you do these cultures with EBV transformed B cells, these cells lack alloreactivity and their risk of GVH is almost nil. And they're otherwise really well set up. And the important point is that with as few as 40 of these, you can cover over 99% of individuals. We've really been able to show this. And the reason for this in EBV and for CMV, but it's not known yet in adenovirus, is that these two latent viruses, when they do reactivate, and throughout our lives when we still have T cells, what they do is they will generate a bunch of T cells early on, but then there's a contraction of these populations. And it turns out that the ones that stay around are T cells that have a very limited number of epitopes that are presented by also a very limited number of prevalent HLAs. And I'll show that clearly in the CMV part of this. We have now transplanted or treated 33 marrow transplant patients and SOT patients. There were 13. This is up through 2015. We obviously, and as Atara now, when they licensed it at that time, they have also treated a large number of individuals. Importantly, these patients had very severe PTLDs, including individuals with CNS disease. They were monomorphic, and they were, as you can see, almost exclusively donor origin. And as you can see, whenever we were able to test them, almost always clonal as well. The first patient that we ever treated here was an individual who had developed an EBV lymphoma in a cord blood graft. And in this cord blood graft, the cord blood cells expressed A1, B8, C7, and they had DRB10301. This patient had received multiple doses of rituximab, but the patient had failed to respond to rituximab. He still had a high level of EBV DNA as shown in orange. What we then did was to give cells from a donor who was homozygous for A1B8DR3. And you can see in the purple the increments each time after the infusions that were given. What you also can see here is that there was a marked level of disease before. Despite Rituxan, the patient had multiple nodes up in the neck and throughout the mesenteric region, and they were also invading the gut, and he was bleeding to the tune of around 2 units per day in terms of the intestines. We gave him the cells, and as you can see, with the cells alone, there was this marked elimination. He completely cleared his EBV lymphoma and continues well today, lives in New Hampshire. Here's a particularly dramatic instance of a patient who developed an EBV lymphoma following a T-depleted graft. And you can again see with one infusion of T cells that this was able within 2 months to basically completely clear the disease. We also find that the EBV T cells, as with the CMV T cells, are capable of getting into the central nervous system. And this is a decided advantage because many of the drugs that are used, including Rituxan, don't get there very well. But as you can see here, a very high proportion of individuals who had issues of CNS EBV lymphomas were able to clear those. As you can see, we had 10 individuals who had either a PR or CR. All of these patients ultimately cleared. Ultimately, the results with the third-party cells look very similar to what you see with unmodified marrow transplants. With the donor-derived T cells, except in the case of the third-party T cell, you usually require more than one cycle. We usually go to 2 cycles. As you can see in the bottom, the ultimate response in the transplant group was 68%, 54% in the SOT recipients, even though they were still on chemotherapy for immunosuppression. Oops, going the wrong way. Each of the patients who responded usually had a quite significant increase also in the peripheral blood of EBV CTL precursors, and that included individuals who had stable disease. In contrast, the patients who had progression of disease either had overwhelming disease at the beginning and had some rise, and there are certain ones who had some rise, but in general, the responses went down. Now, what were the problems? And this is what I'm going to talk about principally today. What are the limitations to virus-specific T cells? And the key variable here are the restraints induced by the HLA restriction of the T cells, because as it turns out, again, with these cultured cells, or even with some of the early cells, what you find in the peripheral blood of normal individuals is a very limited number of T cells directed against the virus, or a limited number of epitopes presented by also prevalent but a limited number of HLA alleles. So, for example, on the top, as you see, the 2 haplotypes A0201, B0702 DR4, A1B8 DR3. Those cells will do very, very well against, if this is the transplant donor, they would do very well against an HLA matched sibling. No problem. That's patient 1 because they're matched. And you don't have to really get nervous about it. However, if you now are doing it for an HLA haplotype disparate individual, and that individual has the typing with the B7, everything is fine if the cells are B7 restricted. On the other hand, if they're B7 restricted and the recipient doesn't have B7, they're useless. And that really becomes the big issue. So the mechanisms that EBV has set in some of these lymphomas is really quite striking. One would be that the EBV lymphoma's origin is disparate from the T cells that you're giving. This holds true for the HCT donor. If it's the HCT donor, almost always the EBV lymphoma will be donor type. If it's host, on the other hand, it's a problem. The third party, you want to be sure you know what the HLA restriction is. Second, EBV itself can downregulate expression of different HLA alleles. It can do so also in an HLA selective way. That's an important feature. And the other key point, that can be either in terms of expression, or it can also lead to degradation of certain HLA alleles. And these are specific proteins of EBV. One of the other ones that we've been surprised at, but we've recently found, is that you can also see in EBV lymphomas, even EBV lymphomas occurring in cord blood recipients, okay? You can see the same thing that you see in recurrence of AML following haplotype disparate transplant, namely the unique haplotype that is allogeneic, okay, to, for example, the host, or in the transplant setting, the donor is deleted. In the case that we found uniparental disomy, the patient had received 2 cord blood grafts. And it turned out that the lymphoma with uniparental disomy deleted the unique haplotype of the cord blood that did not engraft. It's really quite striking. So it was able to live on and do some nasty things. Well, here's an example of a case where the HLA is an issue. This is a young child who had HLH, which was EBV positive. We were worried about EBV, so therefore we made EBV-specific T cells from his haplotype disparate mother. And as you can see in that setting, the mother has uniquely HLA-A1101, and the T cells that were generated, as you can see in the blue, were also HLA-A1101. Those are the T cells that were EBV-specific that we had generated from the mother. He did not, the mother did not develop any cells which were restricted by A2601 at all. This kid, as you can see in the blue, received T cells and continued to have EBV. And as you can see in the bottom one, continued to have this lymphoma growing in the stomach. And this patient received 2 courses of the EBV T cells from mother, and it still continued to regress. What we did in this circumstance was we found in this setting that the lymphoma actually turned out to be derived from the host. So the T cells we were giving had no capacity to recognize that tumor. We switched to a third-party donor whose cells were restricted by A2601. And as you can see in the final 3, that all of a sudden the lymphoma goes away. He continues to live and is a happy young student, one of the brightest kids I've ever met, living in Boston. The other mechanisms are the virus itself. First of all, epitopes that are usually dominant when presented by HLA, by a specific HLA allele, may not be expressed or may be inadequately expressed by the EBV strain in the lymphoma. A second one is EBV epitope is actually mutated, or the EBV epitope is not presented due to impaired processing and presentation. And this can vary by HLA. When we were doing our early work, we found in the spontaneous host EBV BLCLs growing from the tumor, 3 individuals in this cadre who failed to respond. And what we found was these T cells, as you see in the yellow here, the donor CTLs killed B95.8, but did not kill the BLCLs derived from the tumor. On the other hand, if we sensitized with those tumor cells, we could then generate CTLs which were in fact reactive against both B95.8 and the spontaneous BLCL, indicating to us that in fact this patient's EBV BLCL, the dominant antigens, were not expressed on 95.8. Steve Gottschalk presented a really elegant study, again showing in an individual with HLA-A1101 that the population of cells were reactive against EBNA3. And intriguingly enough, in this setting, there was a mutation in the EBNA3 tumor, as you can see, UPN240 tumor. And that mutation led to complete deletion of the antigenic immunogenic sites of that were on EBNA3B that were presented by A1101. This holds true, but we have had other cases in which what we find is we've asked the question, to what degree do different mutations in EBNA3B alter the capacity of the T cells to see the tumor? And as it turns out, the 3B populations of cells, which are reactive, as you can see, against B95.8, they do perfectly fine in blue. But as you can also see, not all of the mutated epitopes actually interfere with CTL killing, but the last one there, the DEPASTEL, that one clearly does. So that's an instance in which you have a mutation where the mutation counts, but not all of these mutations do. The other key thing that has come up, and that's come up in the literature also, is we've had other instances in which the EBNA3B is just not expressed. And you can see in the spontaneous, in orange, down below, the complete lack of the 3B expression and lack of the antigens. although these T cells, which are now again specific for 3B to the exclusion of the others, but this patient does have spontaneous BLCLs, which are able to express other of the latent proteins. This is a particularly interesting other case. This is a patient, again, with HLA A1101. And what we found in this setting is that we gave 3 separate donors T cells to this patient. None of them did anything. And then we ultimately switched to B4403, and we had complete resolution. So this was the first instance in which we could show that actual switch could actually produce a significant antiviral effect. When we looked at this patient for evidence of mutations, we could not find any mutations in any of the peptides again, EDNA 3B that were mutated from B95-8, yet clearly they did not react. The T cells specific for 3B did not react in this setting. So the implication here for us was the idea that, in fact, the epitope was not appropriately presented due to the potential activity of the evasins that EBV has, which are a series of EBV microRNAs, which can interfere with the whole processing and also the movement via tap to the MHC. And this can limit the capacity of these cells to kill off the tumor cell populations. Nevertheless, one of the advantages of having a bank of these cells is that, in fact, you can switch. You can switch to another alternative. And we have found in many of these instances, 6 of 9 instances, that we have been able to then achieve clearance through the switch to T cells that are restricted by a different HLA. Overall, if we now look at the patients who have been treated, who achieved after the initial cycle a CR, they're doing well, 89%. And even those with stable disease at that point, most of those, by virtue of switch and otherwise, we've been able to achieve long-term survival. Overall, if you now look at this, you can see that the incidence of CR and PR for the primary donor and the third-party donor are really quite similar. We're going to talk a little bit about that later on. But here you can see now a large number of groups with small series. We have a significant series of 33 cases, but as you can see, the overall incidences of clearance of CR plus PR ranges for the 40 to 50% on up to 100% for individuals with certified PTLDs, and lower numbers, higher numbers for individuals treated for only viremia. In the CMV thing, I'll go through quickly. In CMV, again, with letermovir, CMV may be becoming a very limited concern. But nevertheless, the incidence of problems with CMV, the reactivation is about the same, matched or unmatched. But when you go to the haploidentical, what you see still is disease incidence is higher and the lethality is higher. In Memorial's experience with T-depleted grafts, it also is shown in the HLA mismatch setting. As you can see, CMV disease 39% versus 12% in the match setting. And again, we are concerned that this may be, again, a function of the fact that the donor's T cells reactive against CMV may not be appropriately restricted for the recipient population of cells. And this is some evidence for that, namely, and we're showing this actually in individuals, but if the donor CMV response is restricted by an allele not shared by the patient, the likelihood of disease is significantly higher. It's 53% in our series. Again, we have a significant bank of these cells. We can now, in this setting, isolate using a matrix system we can isolate the epitope, and we can also then take those epitopes or the whole pool and put them on a series of cells that share single HLAs, and we can thereby show what is the epitope through this matrix and what is the HLA restriction of the cells. You can see on the top that class 1 CD8 population that is reactive against the EDQ epitope, and below the CD4 cells that are restricted by DRB10402. There's also a small population that's restricted by A2601. And we've treated a significant series of individuals, 59 patients evaluable for response, 67 who at some point got a change in the antivirals, so could not be evaluated for response, but only for toxicity. Again, this is to illustrate what I was saying before, namely, there are only 24 alleles represented in terms of the presenting HLAs and 20 epitopes, despite the fact that there is a very, very large number, over 165 different HLA alleles in the A, B, and DR population in our cadre of patients referred for treatment. So again, as you also can see, these are large part very common, like A2, which is the dominant player, a B7, B8, and other ones. There are in our group here individuals who are predominantly recipients of T-depleted transplants, but most importantly, these individuals had received a lot of prior therapy with anciclovir, valganciclovir, foscarnet. The number of prior therapies median is 3. The overall response rates here for patients with viremia alone is 67%, and it's 60% for those with disease plus viremia. Overall, it's 64%. In those who respond, either in complete or partial responses, you see the drop-off in terms of the CMV DNA. In contrast, the non-responders in general just continue to rise. A striking feature, however, in this setting, as opposed to what we found with EBV, was that we couldn't really see significant differences in the number of T cells in the circulation of the responder, non-responder populations. Whether, however, they differ in terms of their quality is another issue that we really need to deal with. Comparison of the characteristics at the baseline that were different for responders or non-responders in the CMV setting. First of all, as you can see, is those who received conventional transplant, 93% of them, or 13 of the 14 patients, responded. 55% of the cord blood plus T cell depleted, or 56% for the T cell depleted alone. So what this really says is if you lack T cells at all in the host, or the graft itself has very little in the way of T cells that are active, you're likely not to be able to have as good a response. What we also can see here is that if you look at the T-depleted transplants, the actual levels of responses that are observed are about a log lower than the responses that you see consistently in recipients of conventional transplants. Intriguingly, however, is number C. Here we're looking at the A0201 tetramers in patients who received T cells that were A0201 restricted. And what there you can see is that there's not much difference between those who got a T-depleted versus those who got conventional graft. Now, this is another important point here, and namely that the prior therapy, we were a little bit shocked at this, but the actual proportion of patients responding who were on ganciclovir as their antiviral, the proportion of those patients responding was lower than what we found if they were on foscarnet or another antiviral. And this may reflect the fact that ganciclovir is a relatively immunosuppressive agent. The other thing to note is that the numbers of CD3, CD4, and CD8, if they're relatively higher, patients do better. And the other one that was really quite striking to us is the fact that when we look at our responses, what we see in these cases is the 66% to 100% A1101 restricted cells always did well. The B8 restricted cells do very well. The others, or at least 2/3 of them, do well. But strikingly enough, when we gave T cells that were restricted by one of the B35 alleles, none of those T cells did anything in terms of clearing the disease. So here we have a circumstance where the B35 restricted T cells failed to respond to the targets. We believe that this is a function of the evasions that CMV has. In this setting, what we found was that the A2 restricted and B35 restricted T cells, when we looked at them in terms of their capacity to recognize targets at very, very evanescent loadings of peptides, as you can see, they're really quite comparable. However, as you can also see in C, in the bottom black line there, the B35 infected CMV cells were not killed, whereas the AO2 infected cells were consistently killed by the A2 restricted populations of cells. So this again is a function of alterations in terms of TAP, in terms of its capacity to take peptides, as well as other a series of evasions that can limit and the processing of peptides such that the peptides never make it that far. And as it also turns out from some really glorious work done by Dr. Amrez over in Munich, which you can also see, is that in very real terms, the effects of these evasions appears to be also relatively HLA selective. So on the right, you have interferon pretreatment. You now look at a series of cells which are carrying specific deletions of evasions like CMV, US11, US2, US3. And as you can see in this setting, if you pre-treat with interferon, you could obviate the responses, the limitations to the responses they now kill. But if you now look out without interferon, you do see the loss of activity. And most importantly, what you see in the wild type for the B3501, almost no killing at all. So this varies. B7 is less effective than B35, and AO201 is significantly affected, but appears still to be relatively squared off on this. And I think this is a big issue for us now to try to figure out what it is about these specific evasions that are relatively HLA selective and to what degree can we overcome them. Nevertheless, the last short thing is to say that the responses are durable. How comes it? If you give third-party cells, you can have these durable responses that last for months to years. This is EBV, as you can see, and this is the CMV. And as you can see in blue, we have only one patient who recurred with the CMV at all, and no patient with EBV who responded died, even though these cells are usually not present that long. So what we think is that, in fact, what is going on is that these populations of T cells will go into sites, will kill cells, and then by cross-presentation can actually induce generation of secondary populations that are endogenous, that are able to sustain the responses. We've seen this, that the third-party CTLs are regularly detected early for about 5 to 6 weeks, but it's rare that they go for more longer periods of time. This is an example of looking at patient after multiple cycles. Here we have one patient who got a kidney transplant who was on tacrolimus, yet we still had evidence of EBV reactive populations of CD137 T cells that were the ones that we were giving from, in fact, the donor of the cells, and they're present 23 months later. We have not found that with CMV. CMV, it's almost always a few of those EBV third-party cells through the first 5 or 6 weeks. After that, it's either the donor's cell populations of EBV reactive T cells, and they're reactive against the same epitope as the cells infused. Or alternatively, you also see, as you can see in the bottom right, the populations of cells that are also derived, that are residual host type cells. As you can see in that circumstance, 56 or 34% of the stem cells from stem cell donor and 66% are from the host. So there are individuals who have this residual population of host T cells that can also contribute to significant protection. So in conclusion, what I can say is that the banked virus-specific T cells are immediately accessible. They can be used in the context of a T-depleted graft. I didn't really talk about this, but if you give these cells preemptively at the time of initial presentation of, or reactivation of these viruses, they can be quite effective. I think Ann Lean and several other groups have shown this. You can identify from the bank a selection of virus-specific cells based on their restriction and also by their epitope. The banks have been applicable to more than 90% of patients, even though the banks may be limited to no more than 40 donors presenting prevalent HLA alleles. You can also use these cells to provide effective switch therapy. And most importantly, these responses are durable. They persist long after the third-party cells have been cleared. This is the group that I've had the glorious time interacting with, and I thank you for listening, if you're still there. Hi.
Rich, that was beautiful. I have many questions, but I will restrict to two. So you mentioned that this third party do not have any alloreactivity. And indeed you showed, because you see some persistence even with tacrolimus up to 6 months. So how do you explain that? Because it's just a matter of HL restriction, or there is more than that?
I think the critical point is that the viral antigens that you have with EBV, CMV, and certainly adeno are really quite immunogenic. And they stimulate a very, very large number of T cells. And so you're literally outgrowing the populations of residual allo cells that might be there. I mean, when you're doing this, this whole system of sensitization is autologous. So you're taking autologous dendritic cells or autologous BLCLs. But the observation that I had in those first slides holds true. I mean, the actual number of alloreactive cells that we're giving is extraordinarily low. It's about 5 or so. In fact, you're probably at sort of the limiting dilution levels that you would see. So I think it's just a function of growth. But that also is one of the reasons why hold on one sec. I want to show you something. One of the reasons why I'm concerned about the multivirus mode as well. Oops, share. Because, oops, wait. Can you see them?
Yes, we see the title.
Okay, hold on one sec. When we looked at this, this slide here was all done by CTL precursor frequencies. Okay. And when we're looking at that, it really is, that's T depletion. Hold on a second. This one here. This is what we got. So this is 5 per million. So if you gave to a 70 kilogram man, you'd be giving 350 potentially alloreactive, major alloantigen reactive cells per kilogram.
But it also means no cross-reactivity. It also means no cross-reactivity between
Correct.
your ABV-specific donor T cells with the host alloantigen, right? So you say that the HLA restriction is dominant on the cross-reactivity with other peptides.
Yes. The T cells that grow out, when we look at our bank, what we find is that if you look at the T cells grown out against EBV transformed B cells, you are usually talking about populations of cells that include maximally 3 populations of EBV reactive T cells directed against 3 separate epitopes.
Yeah.
If you do CMV PP65, the average is around 1 to 2. In CMV PP65, if you have a donor who is HLA B0702, it will always be B0702 to the exclusion of all others. Okay. So they grow out that way, and you have dilution of the alloreactive cells out.
Thank you.
Other thing to be noted, however, that's important is to just make a note, is that in terms of that issue of well, now
It's okay.
I'll skip it.
It's okay.
It's too long. Go ahead.
We got too many other questions. It's okay.
Yeah,
Please,
go ahead.
go ahead.
Hi, Rich. Chris Dvorak here.
Hi, Chris.
So as you know, some of us participated in a trial of multi-virus CTLs run by Mike Keller and Kath Ballard. And that trial was successful, I think, in the post-transplant patients to a degree relatively similar to your data. But where it didn't really work, although the numbers were small, was in the patients with inborn errors of immunity like SCID, who had bad viral infections, and it didn't really work to bridge them to transplant very well. Your data that you presented was mainly in the post-transplant, liquid or solid. Do you have data on patients with inborn errors of immunity pre-transplant that you tried to use these kind of cells?
I'm really glad you're a wonderful straight man. I'm glad you asked that question. Basically, one of the reasons why we think that the populations of cells are basically able to kill the viral infected cells early, but that it's the endogenous populations that are critical long-term is exactly what you're saying. Because what we basically find in this kind of a setting is we found the same. We had one patient who developed an EBV lymphoma who had received years ago, he had DeGeorge syndrome, okay? And he got a marrow transplant. Dick's team gave him a marrow transplant. All right? That's going to supply him with all the immunity that particular donor had, right? But the donor was his younger sister. I'm sorry, his younger brother. That's right. And so he grows up and he gets EBV as a mononucleosis and then develops an EBV lymphoma with DeGeorge. Okay? And we could maintain him for a period of over 6 months by giving him repeated doses of EBV-specific T cells. But he would not make his own T cell response. And I think that that also is really strong evidence for the idea that these populations of cells can really have a significant activity, but their durability is dependent upon the presence of the capacity to make an endogenous response.
Yeah. Thank you, Rich.
And I that's what I expected. And, that paper that you and Kath put together on what happens in the immune deficiency, it's really impressive what happens if you haven't given them a transplant. It's an issue. If you don't have that correction, it's a problem. What we're also now trying to also find out would be, as we get into older and older individuals, because we're transplanting people 70, 75, and you understand that I consider anything younger than myself to be pediatric. So, this whole issue of how do we get around that? And I think, Chris, we're ultimately going to get back to the thymus. We haven't done any studies of doing thymic epithelial grafts since the early study that was done in Seattle, and they were using conventional transplants with post-transplant Psi, et cetera. So, but I think that it may very well be that we're going to need to have either a thymic epithelium or its equivalent, which may be a synthetic thymic mode, which Ben Brink's working on, that would allow, as it were, a new system to grow up.
So Jesse gets the last question.
Okay. Hi, Dr. O'Reilly. This is Jessie Alexander from Stanford.
Hi.
I think this is very interesting, and I think in particular, the area of being able to study some of these things is a little bit easier in the setting of solid organ transplant. In some ways easier, in some ways harder, right? So, so basically on my protocol, which is a bit different because I use the gamma capture technology. But I had I guess in Dr. O'Reilly terms, a quote pediatric, but actually quite older patient who had 2 deceased donor kidney transplants and had very refractory CMV. In fact, had absolutely no drugs left when he got his infusion. The thing that was interesting is, he responded beautifully, but then I would say maybe 9 months later, he had a little bit of viremia, and we didn't draw his blood at that time to say, but he cleared it very well. And so he had a haploidentical donor for his cells. And I'm just very interested to hear your comments on the HLA restriction in the setting of that different methodology.
Well, we've been looking at it at our shop as well now, just to find out a little bit more about it. There are 2 aspects of it. One, the prodigy, if you're isolating the cells, the thought was that you'd have many more younger cells. I'm not sure that that's held. I think when we've looked at them, they're still CD8s and they're still mostly effector memory. You do have a significant number of central memory cell populations. But the other question that also comes up is, with that prodigy system, when you're looking at interferon gamma, is it possible that you are able to isolate small populations of cells so that you don't have that domineering effect over an extended culture period? You're culturing them for what, 24 hours? Are you doing the 24 hours, or are you waiting 3 to 7 days?
No, it's less than 24 hours.
Yeah, it may very well be that, in the circulation, you're still going to have the predominance of the cells that are reactive against the dominant epitope. And that's been shown by Kern when he looked at CMV responses to PP65. He again found that the cells, even at the very earliest stage, would be predominantly, for example, if it's A2, it's going to be NLB, or if it's B7, it's going to be TPR. But you still may have other ones in there, and if you give, a large enough number, perhaps they can expand. The other issue to be raised, though, that I do think is really impressive, is that when I'm looking at some of the experiences that have been published thus far, it turns out that, for example, with these systems, even when you're giving 10 to the third per kilogram, as opposed to, quite a bit more, you can, or I'm sorry, 10 to the third cells, you can still see significant expansions. There are groups using the interferon gamma capture mode who have given doses as low as 10 to 1 to 5 times 10 to the fifth and have had good responses. They see really massive expansion of the T cell populations. Now, that's the other real concern, and that is if you have too much virus or too many infected cells, you're not going to be able to pull it off, or you may not be able to pull it off. Everything in the CMV and everything in the EBV data would say that if you have more extensive disease, for example, EBV with extranodal disease or more than 3 sites, those patients are still at risk even with the cells that you give. Could you potentially do better if you gave really high doses? We haven't explored that, but that's something I think would be really important to do. The one last part of that equation on your end is this disturbing thing that we find with the type of drug used, because ganciclovir is immunosuppressive, and that was really kind of shocking. The degree to which that was a limiting mode. I did not expect that in any way, shape, or form to be a significant player, but as it turned out, it was. So, going with foscarnet or some of the other ones, may make better sense in that regard.
Thank you.
Thank you.